Sequencing both the genome and the transcriptome from the same samples allows you to directly measure the causes and the consequences of complex phenotypes, and this allows you to avoid making inferences when you're interpreting your results. In this video, we'll talk about the best practices for DNA and RNA isolation, library prep, and sequencing so that you can be ready for your first multiomics project.
The great thing about this workflow is that it requires no changes to well-established Illumina whole genome sequencing and whole transcriptome sequencing library prep protocols. The only difference is that we'll make sure that we're isolating high-quality DNA and RNA from the same sample so that they could be directly compared. There are a ton of high-quality kits out there for DNA and RNA isolation, but what's important is that DNA and RNA are isolated together and then split into two samples. One will be treated with DNase to remove DNA, the other will be treated with RNase to remove RNA. The end result is a tube of RNA and a tube of DNA, both from the same sample. We'll repeat this process for all of the samples that will eventually be pooled and sequenced together. Now that we've got DNA and RNA, we're ready to start two separate library preps. Both library preps, however, will share a similar principle. High quality in often means high quality out, so we'll check concentration and quality both before and after each library prep. Let's start with our DNA prep. Illumina PCR-free library prep works by adding DNA to magnetic beads, which are linked to transposomes. The DNA binds the beads and is tagmented. This process involves both fragmentation of DNA and ligation of adapter sequences that make the DNA compatible with an Illumina sequencer. When starting DNA prep, our first step is always to check the quality and concentration of our isolated DNA on a qubit. There are many other tools like nanodrops which are common in many labs, but they're often not quite accurate enough for this use case. After the DNA library prep protocol, that initial concentration measurement will become very important. If your DNA input for the library prep protocol was over 300 nanograms per microliter, a final concentration of each library can be determined using the single-stranded qubit kit, while qPCR should be used for an accurate final concentration if input concentration was less than 300 nanograms per microliter. Now that we've prepared our DNA, it's time for RNA library prep. The protocol can be broken up into three distinct phases: ribosomal RNA depletion, cDNA synthesis, which reverse transcribes a DNA copy of your RNA sample, and finally, addition of adapters and indices to identify each sample and make it compatible with an Illumina sequencer. Like the previous DNA prep, we want to make sure that we have high-quality RNA going into our library prep, so it's a good idea to assess RNA concentration on a qubit, plus to determine the quality of the RNA sample using a bioanalyzer or fragment analyzer. High-quality RNA will show clear peaks for the 16S and 28S rRNAs, plus very few smaller degraded molecules, whereas lower-quality RNA samples will have peaks for degraded RNA molecules and may have some suboptimal results. After you've measured your RNA quality and followed the step-by-step protocol, you've got an indexed library that's almost ready to go. There are a few final checks that we'll perform on our sample before we're ready to load it onto the sequencer. Like our DNA library prep, we'll perform qPCR to quantify our sample. We'll also run the final library on a bioanalyzer or similar instrument to determine if the size matches what's expected from a standard total RNA library prep. By this point, we've completed DNA and RNA library prep, and we've checked concentrations and quality on our final libraries. That means it's time to pool libraries together and sequence. You could determine how many samples that you could sequence by dividing the total number of reads that are possible on an Illumina flow cell by the total number of reads required per sample. In this example of whole genome and transcriptome sequencing, we usually need about 450 million reads per sample. Make sure to check out specific instructions for running DNA and RNA at the same time when using two-color chemistry on Illumina instruments.